Hello, I am here with your video lecture for Chapter 3, Cells. Your next video lecture will be Chapter 5 on tissues. I did not combine them. In the event that you needed information on tissues, you wouldn't have to go all the way through this video lecture in order to access the information that you were looking for. A couple of things that I want to make sure that you have at your disposal while you're looking at the video lecture so that you can go through and make sure that you have them. Number one, make sure you have your textbook. Number two, there are some handouts and documents that I have posted on Blackboard that you may want to access. Number one, the study guides folder is always open and available to you, and you can print off those Word documents at your leisure whenever you'd like to. So make sure that you have the one for Chapter 3. In addition to that, if there are any PowerPoint presentations that are associated with the chapter that I'm covering, make sure you have that. I don't believe that there is a PowerPoint presentation associated with Chapter 3, but if there is, you could have that. And additionally, if there are any documents, now there will be documents that are posted if they're not already posted for this particular chapter. One will be an actual diagram of the cell. And I believe, if I don't have a copy of it right now, I will make sure that it's posted. The information on the cell cycle, yes I do, which looks like this. Okay, so these are the documents that you will want to make sure that you have handy with you. And once we actually fill in the information, I will post it so that you will have it and have accurate information. Or just make sure that if you have any questions in regard to that information that you address me when we convene in class, should we convene. If not, you are always free to email me any of your questions, and I typically can respond within 24 to 48 hours. Okay, the disclaimer, I'm at home. If the dog barks, you know the drill. I'll just let her get it out or try to silence her as best I can, and then I'll pick up from where I left off. So let's get started. Okay, excuse me. A few things just to recap that we have already talked about. Since we are going to talk about cells, we need to remember what the cell theory actually states. The cell theory indicates, number one, that cells are the basic units of structure and function. Number two, all living things are made up of cells. And number three, cells can only come from pre-existing cells. Now, this is information that we covered in the introduction, which was Chapter 1. And from there, we went on to identify the characteristics of living things, and we also identified the levels of organization, starting with the cell, from the cells making tissues, tissues to organs, organ systems, and then the organ systems combined together to make the organism. So we're going to start at the cellular level today. Identify a cell, the basic components of the cell, and their functions. So if you will look in your textbook, beginning with section 3.1 is just general introductory information, so make sure that you do read through that. Section 3.2 actually begins to identify the component parts of a composite cell, just a regular cell. Our body is made up of thousands and millions of cells, actually. And the cells actually have three basic component parts. Number one, we have the cell membrane. Number two, we have the nucleus. And number three, we have the cytoplasm. Within the cytoplasm is when we will find what are called organelles. And the literal translation of organelles or organelle means tiny organ. So as we go through this section, we'll identify the tiny organ and the function of the tiny organ. But first, let's talk about those three component parts that I just identified. Number one, the cell membrane. The cell membrane is very vital and important to the cell because it's literally the first line of defense. Now, there are different types of cells, but we're focusing on animal cells. Animal cells are typically ovoid in structure, meaning they're kind of round. Typical structure of an animal cell. Now, obviously, it has other things inside of it, but that's basically its structure, which kind of differs from plant cells. Plant cells are more rectangular in form, and in addition to that, plant cells have what's called a cell wall. Animal cells lack a cell wall. They only have a cell membrane. So from what I just diagrammed, what the arrow would be pointing to is the cell membrane. The cell membrane has two main functions. Number one, it's the first line of defense from the external environment. So it literally protects the cell. And number two, it acts as a selectively permeable membrane. And what that means is it allows some things to enter the cell while keeping others out. So it's a very important function. The second component that I identified was the nucleus. And I'm just kind of drawing one in. Something like that. Okay. Typically, it's also round in form and in structure. And it's oftentimes, most of the time, it's the largest organelle that you will find inside of the cell. And it's darker stained than many of the other organelles that you'll find in the cell. The nucleus has a very vital role. It's responsible for all of the metabolic activities of the cell, which means basically it controls everything that the cell does. If we had to equate it to an organ inside of our body, it would be like our brain. Our brain is what makes all the decisions. It's the control center, so to speak. And that's what the nucleus is as far as in a composite cell. Now, the third component you actually really cannot see because it's a clear gel-like substance in which we will find all of the other organelles suspended, and it's called the cytoplasm. So as you continue to read that information and you turn the page and you begin to read further into the cell membrane, it goes into a little bit more detail than what it is that we need to know. We just need to know the information that I just presented, which would be number one, that the composite cell is made up of those three component parts, the cell membrane and the fact that it is selectively permeable and it's the first line of defense. The nucleus, which is responsible for all metabolic activity. And three, the cytoplasm, the clear gel-like substance in which you can find all of the other organelles suspended. So you can move through that. There is a diagram of the cell that you see on page 63 or thereabout, depending upon the edition of the book that you have. But it's a little confusing. So I would advise, and we will go through and identify that I look at the cell from this vantage point. You'll notice at the top, I actually have the composite cell. I have some boxes that are not filled in as of yet, but we will go through and do that. And then I have the information at the bottom that literally gives you the name of the organelle and the function, and we will fill those in momentarily. Okay, so if you would then move to the section, we are still in section 3.2, but if you will move to the heading that says cytoplasm, excuse me, it's here that it begins to identify the different organelles inside of the cell. So I'm not concerned that you know about the cytoskeleton and what it's composed of, but we're going to jump right into the different organelles that are listed inside of the cell. So it starts with number one, the endoplasmic reticulum. Now something I want you to do before we get started identifying the different organelles and their functions, even though we have them listed at the bottom of our sheet. If you could please, along with me, you will notice if you have your diagram. If you do not, I will make sure that it's posted and that you have access to it. We're going to start with this box right here. And we're actually going to number the boxes so that you will know where I'm writing information. So let me get a pen that I think you'll be able to see well. Okay. Starting with this very top box, list that one as number one. And then we'll follow it all the way down. Number two is the second box. 3 is the third. 4, followed by 5. Now you will notice that there is an arrow that does not have a box. We do need that arrow, so if you want to extend the line, that's fine. Indicate that as number 6. And then we have the box beneath that, label it as 7. And then finally we have another arrow that does not have a box. Please indicate that box to be number 8, or that arrow. So you should have 1 through 8, and I recognize this may be backwards. I'm sorry, I tried to correct that. But you should have 1 through 8 down this side. Okay, so then beginning on the other side, notice that there is an arrow pointing to a long squiggly thing. We're going to label that as number 9. The box beneath it is 10. The next box beneath 10 is 11. The box beneath that would be 12. The box beneath that will be 13. Now, before you list the next box is 14, we need to indicate another arrow. You will notice that there are dotted lines around this large circular structure. If you could insert an arrow to any one of those dotted lines, that will be number 14. An arrow to any one of those dotted lines around this large structure would be number 14. The box beneath the arrow that we just inserted, please write omit, O-M-I-T, inside of the box. The same thing in that next box, O-M-I-T. And then we will continue to number. The box beneath the second omit will be 15. And then finally, the last box will be 16. Okay. So now we have our diagram labeled. And now that it's labeled, we can continue. So first, looking in the textbook, the first organelle that it identifies is what's called the endoplasmic reticulum. Well, before we do that, I want to identify the structures that we have already talked about. We talked about the nucleus being the control center and typically the largest organelle in the cell. We've also identified the cytoplasm and the cell membrane. So the largest organelle that you see inside of the cell is number 13. Number 13 is the nucleus. Now, what we did not discuss, well, before we get to that, let's go ahead and identify the other components that we've already talked about. Number 7 is pointing to that barrier or that membrane that surrounds the cell. Number 7 is the cell membrane. And I just abbreviated MEM because I'm trying to write large. And then finally, the clear gel-like substance, which is the cytoplasm. If it's clear and it's gel-like, we really can't see it. So the only arrow that's kind of pointing to what looks like nothing would be number 15. Number 15 is the cytoplasm. And again, I apologize if it's backwards. I'm still working on that. Okay, let's go back to the nucleus because there seem to be a few other things that are pointing to something inside or either on the outside of the nucleus. Because the nucleus is so important and it's so vital to the metabolism and the functioning of the cell, it has to have a membrane that surrounds it, that protects it. That is called the nuclear membrane or the nuclear envelope. So number 14 is pointing to what is referred to as the nuclear membrane or nuclear envelope. They are synonymous. So you can write that in now. Okay. Now also, you'll notice that number 12 is pointing to something inside of the nucleus. I'm going to color it in darker because typically this particular organelle inside of the nucleus contains the genetic information, and it's typically darker stained inside of the nucleus. I know years ago we always said the genetic information, be it DNA, which is deoxyribonucleic acid, or RNA, ribonucleic acid, or a combination of the two, was always found in the nucleus. Well, that's accurate because it's actually found within the nucleolus, which is in the nucleus. So either one is fine. But for specific information, the genetic information is found in number 12, which we refer to as the nucleolus. And see, I colored it in a little darker to differentiate. Okay, so we've gotten those out of the way. Let's go back to the textbook and try to identify them as they have them listed here. So the first one is the endoplasmic reticulum. And if you look on your sheet, you will notice that number 4 and number 5, as far as the definitions, not the diagrammatic representation, but the definitions, indicate that there are two types. There is either what's referred to as the rough endoplasmic reticulum or the smooth endoplasmic reticulum. Both the rough and the smooth endoplasmic reticulum have the same function. They are responsible for transporting proteins throughout the cell. These proteins are found in number 2, if you're looking in the textbook, these very round, small round structures that you can either find attached to the endoplasmic reticulum or you'll find them free floating throughout the cytoplasm. They are called ribosomes and they make proteins. So we're going to identify the rough endoplasmic reticulum, the smooth endoplasmic reticulum, and ribosomes. The rough endoplasmic reticulum is the one that actually has ribosomes attached to it. So if we're looking at our diagram, the box that corresponds to the rough endoplasmic reticulum would be number 4. I'm just going to abbreviate it RER because I don't have enough space to write all of that. And then obviously the box beneath it, number 5, is pointing to the smooth endoplasmic reticulum because it does not have any ribosomes that are attached to it. So please go ahead and diagram that now. Write that in now. The smooth endoplasmic reticulum is number 5. And then finally, from the information that I just presented, ribosomes make proteins. And free-floating ribosomes within the cytoplasm are commonly found because as the rough endoplasmic reticulum moves through the cytoplasm, there are ribosomes that will fall off in particular areas, and then there are other ones that will reattach to the endoplasmic reticulum. So number 16 is pointing to a single ribosome that is within the cytoplasm. So please write that in now. Okay, the next organelle that the text begins to identify is what's called the Golgi apparatus. It's also referred to as the Golgi complex. They are synonymous. And the Golgi kind of looks like a stack of plates or a stack of pancakes. But the Golgi apparatus, and if you look at your definitions at the bottom of the diagram, you'll notice it's number 9, and it says it's the packaging site of the cell. Well, it's actually responsible for taking any waste material that may be inside of the cell, packaging it up very nicely and neatly, and then you'll notice number 3 is pointing to something that looks like little round and it's larger than the ribosomes, but little round structures that are directly adjacent to number 11. Number 11 is the Golgi apparatus or the Golgi complex. So you just go ahead and fill that in. And I'm just indicating Golgi. But number 3 works in conjunction with the Golgi apparatus or complex, and they are called vesicles. So if you're looking, it's not actually listed in your definitions that you have on the sheet. However, because they are situated just adjacent to the Golgi, they are called Golgi vesicles, and I'll explain why. And I think if you turn the page, no, it doesn't go into that yet. Okay, so number 11 is the Golgi apparatus or complex. And then number 3 would be Golgi vesicles. Now these Golgi vesicles are actually very small structures that kind of cleave off of the Golgi, and they are responsible for moving throughout the cytoplasm, taking whatever waste material there may be, eventually fusing with the cell membrane to allow that to exit the cell, and then the cell membrane closes back up and the Golgi vesicles move right back near the Golgi. But there are another type of vesicle, and you'll notice that number 1 shows a structure that's very similar to that, and it shows some material that is inside of it that's eventually expelled to the outside. Number 1 is also a vesicle, but it's called a pinocytotic vesicle. I'll spell it. P-I-N-O-C-Y-T-O-T-I-C. Pinocytotic, T-O-T-I-C, vesicle. Vesicle only has one S. Pinocytotic vesicle. Okay. We can continue. So the next, number 4, they're referring to talking about the mitochondria. The mitochondria are responsible for energy production inside of the cell. The energy source is called ATP, which stands for adenosine triphosphate. These look like, they almost look kind of, what's that? Bacillus shape, if you know what a bacillus is. It's kind of like and maybe I shouldn't say that until you get to microbiology, but it's almost like the shape of a hot tamale or a Mike and Ike or something like that. Number 10 is actually pointing to one mitochondrion, but the plural for mitochondrion is mitochondria. So if you could please write that in for number 10. The next organelle that's identified in the textbook are the lysosomes. The lysosomes are represented by number 6. We actually had to draw a line to that. So we're going to indicate that that's a lysosome. And the function of lysosomes is actually to help break down worn out cell parts using an enzyme or using enzymes. Enzymes are those substances that will help to speed up the rate of a reaction. So if there's anything in the cell that should not be there, fragments of cells that may have fragments of organelles that may have broken down, they could disrupt the metabolism of the cell. So the lysosomes are present in order to help accelerate the rate at which the cell can break them down. So lysosome is indicated by number 6. In the textbook, the next organelle that it identifies are peroxisomes, but you can skip over that one. We're not going to identify them in our diagram. Same thing with number 8. It talks about the centrosomes. You don't need to know those as well. But we do have two numbers that we have not identified on our diagram, which are number 8 and number 9. And they actually fall under number 9 in the textbook, cilia and flagella. Cilia are short hair-like projections that are used for locomotion or movement, and they will all kind of beat in a similar direction in order to propel the cell in a particular direction, which differs from a flagellum, which is singular, flagella is plural. It's a long whip-like tail that the cell can flap around to help propel it in a given direction. So number 8 is actually identifying cilia, the short hair-like projections, and number 9 would be a flagellum. Flagella, plural. Now you will notice that number 15, as far as the definitions are on the diagram, indicates what are called pseudopodia. The prefix pseudo means false. Podia or pod means foot. So a literal translation of that word means false feet. And the pseudopodia, even though we don't have it indicated on this diagram, are typically developed by those organisms that don't have, or those cells that don't have either cilia or flagella. Now, there are some cells that have no means of movement at all, but then there are some, and I'll just draw a typical amoeba. Star, come here. Come here. See how well she listens. Star. No. Star. Sit. All right. Okay, so I have drawn, I know, that's my best diagrammatic representation of an amoeba. You can see the nucleus, you can see the cell membrane, and then the clear portion of it would be the cytoplasm. However, because they do not have cilia, or they do not have flagella, they create what are called pseudopods, and the pseudopods are false feet. The pseudopods are created by what is called cytoplasmic streaming. And what happens is this. Ouch. The cell will begin to stream the cytoplasm in a particular direction. If there's some food or if there's something that it's trying to actually get to, it will stream all of the cytoplasm in one direction. In doing so, this area will then elongate. And as it continues to elongate, the rest of the cell will move in this direction, creating what looks like here would be a false foot to help it move in a given direction. So there are actually 3 methods in which cells can move. Either cilia, the short hair-like projections, flagella, long whip-like tails, or the development of pseudopodia, which is created by cytoplasmic streaming. And with that, we have actually covered all of the organelles which you are responsible for knowing. As we continue on in the chapter, it begins in section 3.3 to talk about cellular movement, how different things get in and out of the cell. And there are 2 terms that we need to know. Number 1, diffusion. Diffusion is one mechanism by which materials can enter and exit the cell. Osmosis is another, but osmosis is actually a type of diffusion. So let me explain. Diffusion is the movement of molecules from an area of very high concentration to an area of low concentration. Now, that might not really resonate very well with you, but let me give you a perfect example. If, for example, you were in a closed room with several people, and someone took some type of body spray or some type of cologne. When they initially spray that body spray or that cologne, you can literally see those molecules come out of either the aerosol pump or the spray can or whatever it's coming out of. You can see those molecules. But over a period of time, they dissipate and you can't see them. Well, if you're sitting here, but the person that actually sprayed the molecules is way over there, you're not going to initially smell it when the person actually sprays it from the pump. But over a period of time, those molecules that could first be seen when sprayed will diffuse or move throughout the room until everyone in the room is able to smell it. So literally, those molecules start very condensed to the point where we could see them come out, and they will begin to filter out throughout the space till they reach a point of equilibrium. That's diffusion. Sometimes we have what are called reed diffusers in our homes, where you'll have a liquid in the bottom of a container or some type of jar or some type of glass vase or something like that. They're all decorative. But anyway, you have a very strong fragrant fluid inside of this jar. And you put a reed inside of it, and initially you can't smell it. But what happens is that fluid will travel up the reed until it will eventually release the gas molecules to allow that particular reed diffuser to make the room fragrant. That's diffusion. Diffusion is of any type of molecules. However, if we are speaking specifically of the diffusion of water, that's osmosis. Osmosis is the diffusion of water molecules, only water molecules. And so if you look in your textbook, it gives you an example of simple diffusion, figure 3.11, and it shows how if you have a beaker of water and you take a sugar cube and put it in it, over a period of time, those molecules will disperse throughout the fluid. It's just as if you're making some Kool-Aid. If you make Kool-Aid and you put the Kool-Aid and the sugar and everything in it, well, you can leave it there and let it sit and allow the molecules to eventually diffuse throughout the water. But it's going to take some time. So that's why when we make Kool-Aid or anything like that, we agitate it in order to accelerate the rate at which it diffuses. But if we just left it there, over a period of time, it would eventually diffuse throughout the water. However, if you look at the diagram on figure 3.12, it's showing you the diffusion of some type of molecules there. I believe here it's indicating different types of molecules. So some of them are water molecules, and the other ones could potentially be something like a sugar molecule. But at the top of page 75, it's showing figure 3.15, which is specifically the diffusion of water through a selectively permeable membrane. And you will notice that as the water molecules diffuse from one side to the other, then the water will rise on one side and it will actually get lower on the other. So it's showing you the protein molecules are left behind while the water molecules actually move through the selectively permeable membrane. So diffusion, the movement of molecules, any type of molecule from an area of very high concentration to an area of low. Osmosis is the diffusion of water molecules from an area of very high concentration to an area of low. Okay, so why do we discuss that? Because this is what happens within the cell. You know, if too much water goes in, it could disrupt the metabolism of the cell and the cell could die. If too little water goes in, it could disrupt the metabolism of the cell and the cell could die. So ideally, cells want to be in a state of equilibrium where the amount of water that's going in is the same as the amount of water that's going out. So we have three different types of environments that the cell can find itself in. So I'm going to do my diagrammatic representation of a hypertonic, a hypotonic, and an isotonic environment in which the cell can be in. So these are three cells. And we're just going to say that they're in some water. Okay, so I'm going to explain. Hopefully you can hear me. I'll put this down some. Okay. In this environment, and I'm going to label these A, B, and C so you'll know what I'm talking about. The size of the arrow is indicating how much or how little water is entering the cell. So, in diagram A, you'll notice that there is a little bit of water that can enter the cell, but more water is exiting the cell. In this case, the cell will then begin to shrink. If it shrinks to a point of cellular death, that is called plasmolysis. P-L-A-S-O, P-L-A-S-O, M-Y-L-O-S-I-S. Let me spell it. M-O-L-Y-S-I-S. Plasmolysis. Plasmolysis. Okay, this would be a hypertonic environment because there's just a little bit of water entering cell, but a whole lot of water exiting the cell. Perfect example. This is what happens when we take grapes and take all of the water out of the cell. If we take all the water out of a grape, we get a what? A raisin. And a raisin looks shriveled up. Another example, if you've ever done this, a slug. If there's a slug outside, when I was smaller, my dad would always try to get some salt and put it on it. And the slug would immediately begin to shrink. Because what happens is the salt literally draws the water out of the cells, therefore out of the tissues of the slug. So it begins to shrivel up and shrink. That is a hypertonic environment. Well, ideally, the cell does not want to be in that environment because it could die. Well, the next diagrammatic representation, letter B, is also not a very good situation to be in. Because in this case, lots of water is rushing into the cell, but only a little bit is going out. So this condition would be referred to as a hypotonic environment. So in this case, if a lot of water is rushing into the cell and only a little bit is able to exit, the cell is going to increase in size like a water balloon. The more water that you put in the balloon, the larger the balloon gets. Well, if it gets to a point that the membrane or that the balloon, so to speak, cannot hold any more water, then it will rupture or the balloon will pop. This is called plasmoptosis, and that also results in cellular death. Plasmoptosis. Plasmoptosis. And to me, mop sounds like pop, and that's what would happen. Ideally, the cell wants to be in environment number C, where the amount of water that's going in equals the amount of water that is exiting. That is an isotonic environment. So those are the three mechanisms, or the three different types of environments that cells can potentially find themselves in. And if you look at the diagram, figure 3.16, it's actually showing you a red blood cell. Now, a red blood cell is a biconcave disc. That's why looking at diagram A, it looks like it's a little hollow in the middle. But if we were able to take that same cell and turn it on its side, it would look similar to this. That's a red blood cell on its side, because we can now see the concavity that makes it a biconcave disc? Well, in figure B, you will notice that the red blood cell looks distorted. And it looks distorted because the water is being drawn out of the cell. So that would be the hypertonic example that I showed you. And then finally, letter C is indicating the hypotonic environment, the one in which the cell would actually swell. Okay. The last bit of information that we need to cover, I'm going to put in a separate presentation. And that is because we have a diagram that we need to go over extensively on the cell cycle and mitosis. And I want to make sure that that's very clear. And if you only wanted to access that segment, you can access that segment. And we will also talk about endocytosis, exocytosis, and the different types of endocytosis that cover that. So this does not complete chapter 3, but it completes the information in regard to the organelles and their functions, diffusion and osmosis, and hyper, hypo, and isotonic environments. The next presentation will cover the cell cycle, which includes mitosis, and mechanisms by which the cell will engulf and take in fluids as well as liquids. Have a great day.